So we'll just hold our breath here for 15 seconds or so.
And people are coming on fast.
Yeah, exactly. Bunches of people are logging on now. Perfect, perfect. And Sharon, for your purposes, the YouTube link also seems very popular. There were lots of people streaming during Mark's talk last time, and also people, how many eventually looked at it? 200 and something?
Yeah, as of earlier this week, it was 300 views.
Wow.
Yeah.
Anyway,
It was like a
okay.
pretty constant number. It
Yeah.
wasn't decreasing.
Yeah. So it's 2:00 here Central now, and there's people still logging in, but I think that we should go ahead and start today's Physical Chemistry Division webinar. I'm Christy Landis. I'm at Rice University, and your fellow organizers are, I'm going to name them on their order on my screen. Carlos Baiz, he's from the University of Texas.
Hi.
Rodrigo Noriega, he's from Utah.
Hello.
Anne McCoy, she's at the University of Washington.
Hi.
And our special speaker today is Sharon Hammes-Schiffer from Yale. And what you're looking at on the screen here, in case you haven't been there before, is the Physical Chemistry Division's web page that announces the speaker series that we have. And we're very, very thankful now to have sponsorship from Phys. They have helped us out with funding the webinar registration so that we're going to be able to maintain this webinar in the future. So we're very thankful there to Phys. And the other thing that is important to note on this page, I'm sure you noticed that Professor Hammes-Schiffer is today's speaker, but also we've updated the new Zoom link for the last webinar of the year by Naomi Hollis. And now the new webinar link is there. So if you'd like to attend that, please go ahead and register at the link. How we're going to run things today, in case you didn't come last time, is we are going to curate among us the questions. And so if you have questions during the webinar or at the end, please type those into the chat. And we are going to be organizing those together and taking turns asking your questions for you at the end to Professor Hammes-Schiffer. So just go ahead and type those into the chat feature and we'll get them all organized. And after that, then we will ask all the faculty to go away, and grad students and postdocs who are interested in interacting with Professor Hammes-Schiffer can stay on and get your questions answered perhaps a little bit more personally, as well as we can do here with this online format. And it worked great last time. I'm sure it's going to work great this time. We're trying to find great speakers doing super exciting science to really feature this format and make it nice for everybody. So with that, I'm going to turn it over to Anne McCoy, who is the chair of the Physical Chemistry Division at ACS, to introduce formally our speaker.
Thank you very much, Christy. So today's speaker is Sharon Hammes-Schiffer. Sharon received her bachelor's degree at Princeton and then went on and earned her PhD at Stanford with Hans Andersen before doing a postdoc at Bell Labs with John Tully, who I noticed was on this call, and that's great. Sharon started her academic career at Notre Dame in 1995, and since then has been on the faculty at Penn State and the University of Illinois, and is currently, and since 2018, has been at Yale. Sharon has received numerous honors and awards. She's a fellow of the Biophysical Society, American Chemical Society, American Physical Society, a member of the American Academy of Arts and Sciences, and also the National Academy of Sciences. She was a former Phys chair in 2012, and also served as a senior editor for Journal of Physical Chemistry, was one of the first editors of Journal of Physical Chemistry Letters, and the first deputy editor for Journal of Physical Chemistry B, before moving on to become the editor-in-chief of Chemical Reviews in 2015. Sharon, is known for, as many of you know, for her beautiful work on proton-coupled electron transfer in a variety of different systems, both developing the theoretical tools that we need to look at it, And then I think what today's talk will focus more on is applications to energy conversion and catalysis and more applied systems. She also has a significant research effort looking at the biological application of various types of proton and electron transfer. This work has led to her being the recipient of the 2021 ACS Award in Theoretical Chemistry, and congratulations on that. And today she's going to tell us about, as I said, the work on proton-coupled electron transfer for catalysis and energy conversion. And so you don't want to hear me anymore. I'm going to turn this over to Sharon, and we are all looking forward to your talk.
Okay, thank you very much, Anne. And it's really a great honor to be giving this talk to the Physical Chemistry Division of the ACS. As Anne said, I have many connections to this division and to ACS in general. So today I'm going to be talking about proton-coupled electron transfer in catalysis and energy conversion. So proton-coupled electron transfer, or PCET, is important throughout biology in photosynthesis, respiration, and many different enzyme reactions. And here are some examples. There's a photosynthesis and the enzyme ribonucleotide reductase, which is important, really essential for DNA synthesis. And in both cases, you can see that electrons and protons transfer in different places and in different directions in these very complex processes. PCET is also important in chemical processes relevant to fuel cells, solar cells, and other energy devices. Here's an example of a dye-sensitized photoelectrochemical cell, and an example of a solar fuel cell that splits water into hydrogen and oxygen. And at the heart of it all is PCET. Now I'm going to use a very general, broad definition of PCET. We know that at least one electron and one proton must be transferring simultaneously, but the electron and the proton could transfer between different donors and acceptors, as shown up here, or it could be the same donor and acceptor, which would be hydrogen atom transfer. The electron and the proton can transfer in the same direction, as shown up here, or it could be different directions. It could be completely orthogonal. And the mechanism could be sequential with a thermodynamically stable intermediate, or concerted with no intermediate. And the reaction could involve only one electron and one proton, or it could involve multiple electrons and multiple protons. So all of this falls under the broad definition of PCET. So over the past 20 years or so, my group's been developing a general theory for PCET, which covers all of these different cases. And this theory includes the quantum mechanical effects of the electrons and the transferring protons, and the coupling and the varying timescales among the electrons, the protons, the donor-acceptor modes, the solvent, and the protein. And we've applied this theory to a wide range of different types of systems. Some of those examples are shown here. So in soybean lipoxygenase, shown right here, the electron transfers from the pi backbone of the linoleic acid substrate to the iron of the iron cofactor, and the proton transfers from the carbon of the substrate to the oxygen of the OH ligand. We know this process is concerted because the single electron and single proton transfer reactions are highly endoergic by 20 or 30 kcals per mole, whereas the concerted mechanism is exoergic by about 6 kcals per mole. So it's going to follow the exoergic path and avoid the high energy intermediates. Now this reaction is especially interesting because Judith Klingman's group has studied the kinetic isotope effects, the ratio of the rate constants for hydrogen to deuterium. And they've measured kinetic isotope effects of 80 at room temperature and up to 700 for a double mutant. So they replace one hydrogen with deuterium, and the rate goes down by a factor of 80 or 700. That's an enormous kinetic isotope effect, and it's a hallmark of hydrogen tunneling. And our theory has been able to explain this in terms of hydrogen tunneling and explain the effects of the mutations as well. And I'll be getting back to this a little bit later in the talk. Another example is these proton wires. When you oxidize this molecule here, two protons, these two red ones, will transfer. And in fact, we've designed proton wires to get up to 4 protons to transfer simultaneously. And another example is CH bond activation. When an external oxidant takes the electron from this molecule, the proton transfers to this carboxylate. And an example of heterogeneous PCET is these photoreduced zinc oxide nanocrystals, where an electron transfers from the conduction band of the nanoparticle to this tempo radical in a solution, and a proton transfers from the surface of the nanocrystal to the tempo at the same time. An example of heterogeneous electrochemical PCET is proton discharge to an electrode surface. In this case, there's an acid in solution transferring its proton to a gold surface, forming a gold hydrogen bond. We've also studied photoinduced PCET. And one example is these triads, where photoexcitation leads to this charge-separated state shown here. And the charge recombination process exhibits inverted region behavior. In the inverted region, the thermodynamically more favorable process actually happens slower. So it's the opposite of what you'd expect. And this is important for retaining charge-separated states in energy conversion devices. And we've also studied photoinduced PCET in biology. For example, the bluff photoreceptor shown here, you photoexcite this flavin to a locally excited state, and then an electron transfers from this tyrosine to the flavin, and a proton transfers from the tyrosine to the flavin through this intervening glutamine. And another biological example is ribonucleotide reductase, which involves 6 PCET reactions spanning over 32 angstroms across an aqueous interface. And very recently, Kathy Drennan and Joanne Stubby solved the cryo-EM structure for the active species here, and we're now studying this with our PCET theory. Now, I don't have time to talk about all of these applications in this talk, so I'll be focusing mainly on 4 different systems: the proton wires, the proton discharge to electrode surfaces, the inverted region for PCET in these triads, and then the bluff photoreceptor. But before I get to all of these applications, I want to just give you a feeling for the flavor of the theory so we understand how we use it to analyze all of these different types of processes. And before I even get to PCET, I have one slide on electron transfer. This is just Marcus theory for electron transfer. And of course, many people have contributed to this field. So electron transfer can be described in terms of 2 diabatic states. In the first state, the electron's localized on the donor. And in the second state, the electron's localized on the acceptor. And then the free energy curves for these 2 states are parabolas along a collective solvent coordinate. We have a blue parabola for state 1 and a red parabola for state 2. And the physical meaning of this collective solvent coordinate is shown down here. You can see when the electron's on the donor, the dipoles of the solvent orient themselves in a way that stabilizes the electron on this side. When the electron transfers to the acceptor, the solvent dipoles reorganize to stabilize the electron on the other side. And it's this solvent reorganization from here to here that corresponds to moving from the blue minimum to the red minimum. Now, when the coupling between these 2 states is very small, the reaction is non-adiabatic, and the rate constant is given down here. You can see it depends exponentially on the free energy barrier, which can be expressed in terms of delta G naught, the free energy of reaction, and lambda, the reorganization energy. And so we just have this simple Marcus expression here to relate all of these to the free energy barrier. The rate constant is also proportional to the square of the coupling in this regime. Now, this is just Marcus theory in a nutshell. Casey Hines and coworkers have extended these concepts to proton transfer, where now these 2 states correspond to the proton localized on the donor or the acceptor. And instead of electronic states, these 2 parabolas are proton vibrational states. But otherwise, the physics of the problem is pretty much the same. So what we did is we said, let's take ideas from single electron and single proton transfer and combine them to develop a theory for proton-coupled electron transfer, where now we have an electron and a proton transferring. So now instead of 2 states, we need 4 states. In the initial state, the electron and the proton are both on their donors. And in the final state, the electron and the proton are both on their acceptors. And now we have 1 state where only the proton is transferred and 1 state where only the electron is transferred. So this framework allows us to describe both sequential and concerted mechanisms. Sequential corresponds to going along the outside of this rectangle. We could have ET-PT or PT-ET. And concerted corresponds to going along the diagonal. We call that EPT. And the mechanism is determined by the relative energies and couplings among these 4 states. When the off-diagonal states are much higher in energy, then the reaction is going to follow the diagonal to avoid those high energy states. And that's the case that I'm most interested in, concerted PCET. So in our theory of concerted PCET, we still have a reactant where the electron's on the donor and a product where the electron's on the acceptor. And the free energy curves are still parabolas along a collective solvent coordinate. And here I show only the lowest 2 parabolas, but in fact, we have 2 sets of these parabolas corresponding to the different proton vibrational states for each electronic state. So you see we have a blue set for the reactant and a red set for the product. And the splittings between these parabolas, those are just the splittings between the proton vibrational states for the reactant or the product. Now it's easier to just look at one pair, so we'll look at the lowest pair here, and we need to think about what the proton is doing. So for the reactant, where the electron's on the donor, the proton donor well is lower in energy, and the proton's localized on the donor side. For the product, where the electron is localized on the acceptor, the asymmetry of that proton potential flips. Now the acceptor well is lower in energy, and now the proton is localized on the acceptor side. And this is just due to the electrostatic attraction between an electron and a proton. Whichever side the electron is on, it's going to pull the proton onto that side. And as we move along this collective solvent coordinate, we change the relative energies of these two states. So at the crossing point here, they're degenerate. And this is the point where the electron and the proton are both going to tunnel simultaneously from their donors to their acceptors. So putting this in movie form, the system starts out with the electron and the proton on their donors. Now, reorganization of the environment leads to this crossing point. Now the electron and the proton both tunnel simultaneously from their donors to their acceptors, and then further reorganization of the environment will stabilize the product state. So that's the general picture that comes out of this PCET theory. Now, typically PCET reactions are non-adiabatic because of small vibronic coupling, so we can use Fermi's golden rule to derive rate constant expressions. And here's the simplest one. You can see it looks kind of similar to Marcus' theory for electron transfer, but there are some important differences. First, you see these summations over mu and nu. These are summations over the reactant and product vibronic states, these proton vibrational states, the blue set of parabolas and the red set of parabolas. Each term depends exponentially on a free energy barrier, which can still be expressed in terms of delta G naught and lambda for each pair of parabolas. Each term also depends on the square of a coupling, but now this is a vibronic coupling. And the form of this coupling depends on what we call electron-proton non-adiabaticity. But in the electronically non-adiabatic regime, it's just the product of an electronic coupling and this overlap S between the reactant and product proton vibrational wave functions. That's shown here, this purple region here, that's S, that's the overlap. And that plays a very important role in determining the rate constants and kinetic isotope effects of PCET reactions. And that overlap also depends very strongly on the proton donor acceptor distance. We'll call that capital R. Clearly, as we pull R apart, right, we increase R, so we pull the donor and acceptor apart, that overlap will decrease. Now here's an example of a molecule where when we oxidize it, we take out an electron, the proton transfers from this oxygen to this nitrogen. So in this case, R is the oxygen-nitrogen distance. And you can see from this normal mode, this is just a thermal fluctuation, that this thermal fluctuation and others will change the oxygen-nitrogen distance. And we need to include that motion in our theory. And we've done this in two different ways. First of all, we derive analytical rate constant expressions, where we approximate the overlap is decreasing exponentially with R. But then we also used an alternative thermal averaging procedure, where we calculate the rate constant at different values of R, weighting by the probability of sampling that value of R, and then integrating over all R. And we've shown mathematically that these are rigorously equivalent in certain well-defined regimes, but the thermal averaging procedure turned out to be more general, more robust, because it avoids this approximation of the overlap decreasing exponentially with R. So this is circled in red because this is what we're going to be using for the examples I'm going to talk about today. Now we've also extended this theory to electrochemical PCET. In this case, the electron transfers between the solute complex and an electrode, and a proton transfers within the solute complex. So the physics of the problem is pretty much the same as what I've already talked about. When the electron's on the molecule, the proton donor well is lower in energy. And when the electron transfers to the electrode, the asymmetry of this proton potential flips, and now the proton is localized on the other side. And we've derived expressions for the electrochemical rate constants and current densities. And it looks pretty much similar to what I showed you before, except that now delta G needs to depend on the applied potential to the electrode. And we now need to consider a continuum of states in the metal electrode. You can see this red continuum here. And we need to consider the electrical double layer effects. What's happening with the solvent and ions at this interface between the electrode and solution? And this theory now can cover both homogeneous and heterogeneous electrochemical PCET. In homogeneous electrochemical PCET, the electrode is just playing the role of a reservoir. It can take electrons and it can give electrons, but it's not involved in the chemistry itself. And some examples are shown down here. This is a nickel P2N2 complex. The electrode gives an electron to the nickel, and then a proton transfers from this pendant amine nitrogen to the nickel, forming a nickel hydride. In this example of proton wires, we remove an electron and 2 protons transfer. In both of these cases, the proton transfer is happening within the molecule. The electrode just gives and takes electrons. Now, in heterogeneous electrochemical PCET, the electrode is involved in the chemistry itself. For example, if a proton is transferred to the electrode, it forms a metal hydrogen bond. And the zinc oxide nanocrystal I talked about before is an example of heterogeneous PCET, because while the electron is coming from the conduction band of the nanocrystal to the tempo, the proton is coming from a surface oxygen to the tempo. An example of electrochemical heterogeneous PCET is the proton discharge to an electrode surface, where an acid gives a proton to the gold, forming a gold hydrogen bond. And I'll be talking about one example of each, the proton wires first, and then I'll get to the proton discharge example. Now, the proton wire application is inspired by photosystem 2, which splits water into hydrogen and oxygen. Now, in photosystem 2, an electron takes P680 here, takes an electron from tyrosine, which then transfers its proton to histidine. And it's been hypothesized that then the histidine gives its proton to something else, and there might be a proton wire. It's not clear that this specific proton wire exists, but we know that in bioenergetic systems, proton wires are very important, right? We need to pump protons across a membrane to create a proton gradient. And this is important in bioenergetics. So we want to figure out, can we follow nature's example and assemble a proton wire where instead of P680 here, we use an electrode to take the electron, and we can get 1 proton to transfer, or 2, or 3, and so forth, forming a proton wire to transport protons over long distances. And this is a collaboration with Tom and Anna Moore, and they've been developing artificial photosynthetic systems where they represent this histidine-tyrosine pair with a BIP, or BIP molecule. In this BIP molecule, when you remove an electron, the proton transfers from an oxygen to the nitrogen here. And that's all that can happen here, because you see there's a hydrogen here, so really this is it. So the question in this project is, can we replace this hydrogen with something that would take this proton to get a second proton to go, and then keep building it out in a proton wire? And we want to do this while retaining the desired oxidation potential of 1 volt versus SCE, because it needs to fit into our artificial photosynthesis system. It needs to be able to split water. And our calculations have been guiding the design of these kinds of proton wires. So we started out looking at the 2 molecules that they had already made in the lab. So one is the parent molecule, which, as I already said, it's got a hydrogen here, so only 1 proton can go. So we can use the square diagram I showed earlier, and we can look at the thermodynamics. And what we find is that the concerted mechanism is thermodynamically favorable by around 7 kcals per mole. It's going to follow the diagonal. We also calculated the kinetic isotope effect, just this ratio of rate constants for hydrogen to deuterium, and got a value of 1.9. In this case, there's no experimental value for comparison. But then we looked at the molecule that they had designed to take the second proton. And when they gave it to us, they actually thought the second proton was transferring. So the question we wanted to answer is this a 1 electron, 1 proton transfer reaction? We call it EPT. Or is it a 1 electron, 2 proton transfer reaction? We call that E2PT. And in order to do this, we need to actually go beyond a square diagram. Because if we have n charges transferring, we need 2 to the n states. In this case, we have 1 electron and 2 protons transferring, so that's 3. So we need 2 cubed states, we need 8. So we need to, instead of a square, we need to look at a cube. And if both protons are transferring, the 2 shown here, then we would be going from this 1 AA corner all the way to the 2 BB corner. That corresponds to electron and 2 proton transfer. But when we look at the thermodynamics, we see that we stay on the front face of this cube. It only does EPT. It would be actually thermodynamically unfavorable for that second proton to go by 14 kcals per mole. So it's not going to go, it's stuck. So we determined that this is indeed just plain old EPT. The second proton is not going. We also calculated the kinetic isotope effect to be 2.1, and that's in agreement with the experimental value of 1.8. So we see that both of these systems are just plain old EPT. They both have redox potentials of around 1 volt and kinetic isotope effects of around 2. So that was a little disappointing because we wanted that second proton to go. So what do we need to do? Well, what we need to do is increase the pKa of this substituent to promote the second proton transfer. And so we did that by designing molecules on the computer before they had even made them in the lab. So we designed these 4 molecules. And the first 2 we found still did EPT, but the ones in gray here, they did E2PT. You can see from this cube, we're going from 1AA to 2BB. So we're getting that second proton to transfer. But at the same time, we noticed the redox potential shifted. It went from around 1 volt down to 0.6 or 0.7 volts. So we see that the E2PT process shifts the redox potentials to less positive potentials by around 300 millivolts. We also calculated the kinetic isotope effect and found that the E2PT systems had kinetic isotope effects of around 1.3, and that's lower than the kinetic isotope effect of around 2 that we got for the EPT systems. So we made two predictions. We predicted that these E2PT systems would shift the redox potential by 300 millivolts and decrease the kinetic isotope effect from 2 to around 1. Then they made these molecules in the lab. And what you can see here is that it went from 1 volt down to these 0.5 to 0.6 volts. So we see this shift. You can see it in the CVs. The blue CVs are EPT and the red ones are E2PT. We see this shift of 300 millivolts downward. They also measured the kinetic isotope effect to be around 1, and that's in agreement with our prediction of an isotope effect of around 1.3. So both of our predictions were validated experimentally, the shift in the redox potential and the decrease in the kinetic isotope effect. But now I want to go on just a little tangent here, because this kinetic isotope effect of 1 might be kind of confusing for people, because often experimentalists use the kinetic isotope effect as a litmus test to determine is the proton going or is it not. And if they see a kinetic isotope effect of 1, they would say the proton's not transferring. But in this case, we know it's transferring. We see the two protons transferring. So in order to understand this within our theoretical framework, we need to think about the excited vibronic states. Remember the stacked blue and red parabolas. So here, just to simplify it, I show only two reactant parabolas and one product. And these two reactant parabolas correspond to the ground proton vibrational state and the first excited proton vibrational state. You can see that here in the proton potentials. And so we have two different pairs of states, the 0, 0 and the 1, 0. And the relative contributions of these pairs is determined by a balance among three different factors. First, we have the Boltzmann population, which will favor the ground vibrational reactant state. And second is the free energy barrier, just the difference between this blue minimum and its crossing point. And you can see that that's going to favor the 1, 0 pair. And then we have the vibronic coupling, which is related to the overlap, and that's going to also favor the 1, 0 pair because of the delocalization of the excited vibrational state. So it turns out that the balance among these different pairs of states, and usually you have more than two, you have to have all the stacked parabolas, that's going to be determined by a complex balance of these different factors. But for each pair, we know that the kinetic isotope effect is going to be the square of the hydrogen overlap over the square of the deuterium overlap. That's just the prefactor in the rate constant expression. And we know that this ratio is going to increase as the overlap decreases. You can just show that mathematically. So what does that say then about soybean lipoxygenates, where I said that has a huge kinetic isotope effect of 80 or even 700 at room temperature? Well, in this enzyme, we found that the 0, 0 pair dominated. So ground-to-ground state tunneling was dominant. And it had very small overlap. And this explains why the isotope effect is so big, because this ratio of overlaps is huge. And the reason for this is that the proton is transferring from a carbon to an oxygen, and that's a weak hydrogen bond. And it's also occurring with a constraint of the active site of the enzyme, so it's not allowed to align very well. And in fact, in the mutant, it goes up to 700, the alignment's even worse, and that's why the overlap is smaller and the kinetic isotope effect is even bigger. So we can explain these huge kinetic isotope effects simply in terms of the overlap being very small. And this tends to happen in constrained environments. That's when you see these huge kinetic isotope effects. Now back to these bit molecules. So for the bit molecule that does EPT, we find that the 1, 0 pair is dominant. So if you look at these overlaps, you see you're going to have intermediate overlap. You're going to have a kinetic isotope effect of around 2, just a moderate kinetic isotope effect. We've also looked at bit molecules with a porphin attached to it. The proton potentials change in a way that now the 0, 1 pair is dominant, and the overlap's nearly unity, and so you're going to get a kinetic isotope effect of around 1. And that's what's happening with the E2PT systems. Here you have to actually look at two-dimensional proton potentials, two-dimensional wave functions, so it gets a little complicated. But the physics of the problem is that the excited states with large overlap are dominating, and that's how you can get a kinetic isotope effect of around 1. So what we see from this slide is that in PCET, you can have kinetic isotope effects ranging from anywhere from 1 all the way up to 700, depending on which excited vibronic states are contributing and what their overlaps are. So going back to the bit molecules, remember we had two predictions, right? We predicted that there would be a shift in the redox potential by about 300 millivolts, and we predicted that the kinetic isotope effect would decrease from 2 to 1. And both of those predictions were validated experimentally, but we still didn't really prove that that second proton was going. So our experimental collaborators said, let's take the E2PT system that we know has the shifted redox potential, this red CB, and let's add trifluoroacetic acid to protonate this exocyclic amine. so that you block the second proton transfer. So it can only do EPT, it has no other choice. And then we see that the redox potential shifts right back up to 1. So this is evidence, pretty strong evidence, that this second proton transfer is responsible for the shift in the redox potential downward. And if you still don't believe it, then spectroelectrochemical experiments also verify that second proton is transferring in these immunosubstituted systems. So we know absolutely, definitively that it is transferring. So we started with these EPT systems, right, where we only had 1 proton transfer, and we then designed these E2PT systems where we got the second proton to transfer, but we paid a cost, right? We paid 300 millivolts reduction in redox potential. So now we're out of range. You see these blue circles here? We need to stay within 0.8 to 1.3 volts to split water. So we overshot. So then what we said is, okay, let's go back. Let's make the pKa not so large. Let's use imine substituents. And then we got right in range. These white circles here, about 0.93 to 0.97 volts. Now we are able to split water with these kinds of molecules. We can also tune the redox potential with a substituent R here. You can see there's actually a linear correlation between the redox potential and the Hammett constant of these substituents. Of course, the electron withdrawing and donating properties of this R group will change the redox potential. But more interestingly, it actually can change the products. We actually get a mixture of E2PT and EPT products for some of these. And so if you look at the equilibrium constant, the ratio of the concentration of E2PT to EPT products here, you can see for the cyano substituent, we have almost all EPT products. From a methoxy substituent, we have almost all E2PT products. So what this tells us is that there's a substituent that's like 12 angstroms away, and that's able to change the redox potentials and the products of the oxidation of this phenol. And so that kind of control can be actually very useful for catalyst design. But so far, we've only gotten 2 protons to transfer. What if we want more protons to transfer? And so we designed a series going from E2PT to E3PT to E4PT. So we've gotten 4 protons to move reversibly over 16 angstroms across this hydrogen bonded network. And you can see they're reversible. These are the CVs, the blue, pink, and the green. They're beautifully reversible. Our experimental collaborators made these. And so we can basically reversibly make these protons go up and down this hydrogen bonded network. And then the question is it actually concerted? Now, in the IR spectroelectrochemistry, they only see all the protons on their donors or all on their acceptors. They never see an intermediate with the IR spectroelectrochemistry. With our calculations, we can calculate the redox potentials to be with only 1 proton, only 2, or only 3, or all 4 protons transferring. And the value that matches experiments is when all 4 protons transfer. So that's evidence that thermodynamics says that all 4 protons are transferring simultaneously to get the redox potential they measure experimentally. So on the electrochemical timescale and from thermodynamics perspective, it appears that these are simply concerted. They're all going at the same time. But what happens if we look on a shorter timescale, if we look on the ultra-fast femtosecond timescale? And the way to do this would be to attach a photosensitizer to this BIP molecule. And then we can actually study, instead of electrochemically inducing proton transfer, we can photochemically induce proton transfer just by photoexciting this photosensitizer. And to study this, we equilibrated the system in the ground state, and then we just instantaneously remove an electron. And we do non-equilibrium first principles molecular dynamics simulations. And this is a movie of one of our trajectories. We ran hundreds of trajectories. And what we found is that the reaction is concerted in the sense that there's no thermodynamically stable intermediate, but it's asynchronous in the sense that the first proton transfer happens about 100 femtoseconds after electron transfer, after we remove the electron. And the second proton transfer happens about 600 femtoseconds after the first one. You can see that in the movie. This is the first proton transfer. This is the second one down here. And you can see that there's a delay of around 600 femtoseconds. So we call this asynchronous in the sense that they're not going exactly at the same time. Now, you're not going to see this kind of asynchronicity on the electrochemical timescale, but you can see it if you do ultra-fast spectroscopy. And so this is a prediction, and we're waiting to see if spectroscopically this kind of asynchronicity can be observed for these kinds of these sorts of proton wires. In fact, they've already attached a photosensitizer to it, so these experiments are well on their way. So this is an example of why the language actually ends up being kind of important, because concerted or asynchronous, it gets a little bit complicated, especially for photo-induced reactions, because in some sense, there is no, it's out of equilibrium when it's photo-excited. So whether it's concerted, whether it's a stable thermodynamic intermediate, it becomes almost a semantic issue, but clearly you see this level of asynchronicity in this particular molecule. So next I want to move to a heterogeneous PCET example. And the example I'm going to look at is PCET from triethyl ammonium acid to a gold surface in acetonitrile. It's important that it's in acetonitrile, not water here. So what's happening is the proton is transferring, forming this gold hydrogen bond. And this step is rate limiting for the hydrogen evolution reaction. We became interested in this because experiments from Yogi Surendraneth's group show a potential dependent kinetic isotope effect. In other words, if they plot this log of the current density as a function of applied potential, they get different slopes for hydrogen and deuterium. You can see there's a different Toffel slope, there's a different transfer coefficient. And this leads to a range of kinetic isotope effects. In this case, it's 1.5 to 6.3 over this range of applied potential study. And this is surprising because electrochemists often use the Toffel slope as an indication of mechanism. But in this case, you have different Toffel slopes, but the only difference is hydrogen versus deuterium. In our PCET calculations, we're able to reproduce these results and explain them in terms of different contributing excited vibronic states for hydrogen and deuterium, and these contributions depend on the applied potential. So in order to understand this, we had to extend our theory to the case of heterogeneous PCET, where now the proton is transferring to the gold surface, forming a gold hydrogen bond. And now the rate constant depends on the distance of the acid from the electrode, this is R, and the applied potential E. You can see the expression looks similar to what we saw before. We still have the summation over the reactant and product vibronic states, those stacked parabolas. But now we're integrating over the one electron states of the electrode, weighted by the Fermi distribution and the density of states. We still have the square of the vibronic coupling, but now we need to use a general form of this vibronic coupling that spans the adiabatic and non-adiabatic limits. We also need to worry about what's happening in the electrical double layer, because in this case, PCET occurs in the inner Helmholtz layer, right? It's got to get close enough to the electrode to give its proton to the electrode to form that gold hydrogen bond. And we know that in this inner Helmholtz layer, there's a drop in the electrostatic potential. It's typically assumed to be around linear drop. And so we developed an extended Gooey-Chapman-Stern model, it's just a multi-layered dielectric continuum model, to describe this linear drop in the potential, and then we include that in our delta G. So now our delta G is the sum of the free energy difference between states, the applied potential E, the energy of the one electron state, and all of the electrostatic and non-electrostatic work terms due to these electrical double layer effects. Once we have all of that in there, we can get this rate constant, and then we get the current density by integrating this rate constant over R, weighting by the concentration of this acid AH plus at each different value of R, at each distance from the electrode. When we do all of that, we get this expression, we get these results shown here. You see here's experiment and here's theory. And you can see that we were able to match the experimental results of the potential dependent kinetic isotope effect, right? The different Toffel slopes, the different transfer coefficients. Now it's important to realize that in this case, we had some free parameters. We had several free parameters that we could use to fit the experimental data. In the proton wire example, all of the input quantities to the rate constants were from DFT calculations and other computational methods. There were no free parameters there. But here we do have some free parameters, but they're physically reasonable values. And more important than getting the exact quantitative numbers correct is that we got a physical explanation for why you would see different Toffel slopes for hydrogen versus deuterium. And the physical explanation has to do with different pairs of proton vibrational states dominating for hydrogen and deuterium with a different dependence on the applied potential. So we can go back to these stacked parabolas I showed before. And remember, one of the factors that determined the relative contributions of the different pairs of states is the Boltzmann population. And the Boltzmann population favors the ground state, but you also have contributions from the excited reactant states. And so because the energy level splitting is smaller for deuterium than for hydrogen, we're going to have more contributions in the excited reactant vibrational states for deuterium than for hydrogen. And we can actually see that if we plot the percent contribution as a function of applied potential. You can see for hydrogen, we have the 0, 0 and the 1, 0 states contributing. But for deuterium now, the 2, 0 state starts to contribute. And that's because the energy level splittings are smaller for deuterium, so it can access the second excited proton vibrational state for the reactant. And if we then plot each one of these, the log current density as a function of applied potential for each one of these pairs, we see that the 2-0 pair has a smaller slope. And that just has to do with the fact that it's less endoergic, and you can actually look at the map and see where that would come from. This is the transfer coefficient over here. But the most important concept to take home here is that when you then average over all of these different contributions, you get the smaller slope for deuterium than for hydrogen. And so this whole explanation here relies on the fact that we are treating the transferring hydrogen nucleus quantum mechanically and including excited electron-proton vibronic states. And as far as I know, there's no other explanation for these experimental results. So you really do need to include these kinds of nuclear quantum effects in order to understand certain phenomena in electrochemical PCET. Now I want to switch gears and talk about photoinduced PCET, the Marcus inverted region. So in the inverted region, the rate constant decreases as the reaction becomes more thermodynamically favorable. In other words, as delta G naught becomes more negative. And Marcus actually predicted the inverted region for electron transfer way back. And this is his expression that I showed earlier. And if you plot the log of the rate constant as a function of the driving force, you see that in the normal region, as you bring this red parabola down, that corresponds to increasing the driving force, you're decreasing the free energy barrier, right? The free energy from the blue minimum to the crossing point. You're bringing the red parabola down, that's getting smaller. And so the rate's going to get faster. And then you get to the point where it's activationless, right? There's no free energy barrier at all. It's zero because negative delta G naught is equal to lambda. You can look at this expression and see that you'll have zero free energy barrier. Now, as you continue to bring the red parabola down, you're in the inverted region. You need to climb up here, the other side, and the free energy barrier starts getting bigger again, which means the rate constant is going to start going down. And that's what's seen in this picture here. And it was actually seen experimentally by Claus and Miller. And that's one reason, the main reason that Marcus won the Nobel Prize, is because he predicted this inverted region behavior. So what about PCET? Well, back in 2009, we predicted that the inverted region might not be observed for PCET because of these excited proton vibrational states. Remember, we're summing over those stacked blue and red parabolas. So you can see here that we have a single reactive blue parabola, but now we have a whole set of product parabolas. And if we only include the lowest red parabola here, then we will see inverted region behavior. This dashed line shows it, exactly how we saw it for electron transfer. But if we include the whole stack of parabolas and we bring the whole stack of parabolas downward as we increase the driving force, then what happens is the bottom one, say the lowest parabola, goes into the inverted region. The next one becomes activationless, and so it takes over. And as you continue to bring the whole set down, then the next one will become activationless. And there's always another parabola there. So you might imagine there would be a plateau. And this is seen for some systems, but on the other hand, for others, our collaborators, Jim Mayer and Leif Hammerstrom, observed the inverted region for this anthracene-phenol pyridine triad. So in these experiments, they photoexcited this anthracene to a locally excited state, and then they observed charge separation. They electron transferred from the phenol to the anthracene and a proton transferred from the phenol to the pyridine. This led to this charge separated state here. And then there's another PCET reaction, which is charge recombination, where the electron and the proton both go back to where they started. And they looked at a whole series of different substituents, 1 through 8 here, where R1 and R2, these substituents on the anthracene and pyridine, are different. And what they found is that for the charge separation state, first PCET reaction, they found that everything is pretty much behaves normal. It's in the normal region. But for charge recombination, they observed inverted region behavior. You can see here that the triad 1 is thermodynamically more favorable, yet it's slower than triad 3. And so then the question is, how do we explain this with our PCET theory? So we had to do a lot of DFT calculations, other types of calculations on these triads, and then we analyzed our results. So in this case, it's charge recombination. So we start with one blue parabola. This is the charge separated state. We found that the other ones we didn't need to include. And then we are going to the ground state. And so we have a whole set of red parabolas. Here we show only states 3 through 7. And what we found in our analysis is that the 0-3 transition dominates. This one right here. And this is in the inverted region. You can see that to go from the blue minimum, you have to go up the other side here, right, to get to the 0-3 transition here. And it also has appreciable overlap between the proton vibrational wave functions. And so it makes a significant contribution to the rate constant. Now you might ask at this point, wait a minute, what about the 0-7 pair? Shouldn't that actually be dominant? Because it has no, it's activationless. It has no free energy barrier at all. right? And that's true. It has very, very small free energy barrier, almost zero. But also, if you look at the 7th excited proton vibrational state, you see it's highly oscillatory. And so you have actually phase cancellation with this blue reactant proton vibrational wave function. So it turns out you have negligible overlap between these proton wave functions, and this pair of states is prohibited from contributing to the rate constant. So yes, you have a zero free energy barrier, but it's also multiplied by zero, a prefactor of zero, so it doesn't matter. It's prohibited, and all the ones above it are also prohibited from contributing to the rate constant. So what we learned from this is that the inverted region behavior will be observed under the following two conditions. First, the state pairs that have significant overlap need to feature negative delta G naught greater than lambda, like the 0,3 pair. They need to be in the inverted region. Second, the state pairs that are nearly activationless need to feature prohibitively small overlap, like this 0,7 pair. So in other words, the vibrational wave function needs to be oscillatory and above the barrier. So it's crossing, the whole proton potential, and it's going to lead to this phase cancellation. If those two criteria are satisfied, then you can see inverted region behavior. We also predicted that the kinetic isotope effect for this system would be unity because of delocalized proton vibrational wave functions. And in fact, they saw experimentally that the charge recombination exhibited a unity kinetic isotope effect. And so that's another example of how we know proton transfer is happening, yet the kinetic isotope effect is 1. So in this case, we were allowed to use our analytical rate constant expression for this system because the charge separated state is fairly long-lived. It lives several hundred picoseconds in some cases. So that means that the solvent can equilibrate and everything's at equilibrium before PCET happens. But we're also interested in the non-equilibrium dynamics of the forward PCET. In other words, if we equilibrate the system in the ground state and instantaneously photoexcite it to this locally excited state of the anthracene, then we're going to see electron transfer from the phenol to the anthracene and proton transfer to the pyridine. And we want to study this non-equilibrium excited state molecular dynamics. We did this using TDDFT. So we photoexcited, in this case, to this S1 state, which is the locally excited state. And then we saw that this state transitioned to the charge separated state. And that's where electron transfer happens. And then we saw that proton transfer happens very quickly after electron transfer. So you can see that here. This is the movie. You can't really see the electron transfer, but you see the proton transfer part, and it happens within a few femtoseconds of electron transfer. So this is a case of concerted but synchronous PCET. That's different from the proton wires where it was concerted but asynchronous. We also were able to use these non-equilibrium dynamic simulations to explain why only certain triads exhibited inverted region behavior. So we looked at triads 1 and 3, which exhibit a long-lived charge-separated state and inverted region behavior. And we also looked at triads 4 and 6, which do not exhibit that behavior. And so these are the definitions of these 4 triads. And so I already showed you the results from the triad 1, where you photoexcite to this S1 state, right? You go to the locally excited state, and it transitions to the charge-separated state, right? The electron transfer to the anthracene. But when we looked at triads 4 and 6, we saw different behavior. When we photoexcited to the S1 state, now it transitioned to what we call the LEPT state. That's the local electron-proton transfer state. In this case, the electron actually transfers to the pyridine, not the anthracene. And so it's sort of like a hydrogen atom transfer from phenol to pyridine. And so the electron can go 2 different directions. It can transfer to the anthracene, which would lead to the charge-separated state, or it can transfer to the pyridine, leading to this LEPT state. And the substituents will stabilize one or the other. So when the substituents stabilize the electron on the anthracene, then you're going to see the long-lived charge-separated state and inverted region behavior like triads 1 and 3. And when it doesn't, then you won't see this behavior. So this allows them to design different kinds of molecules to see the inverted region behavior. And so we just predicted the existence of this LEPT state, which would provide an alternative relaxation pathway. But my understanding is that Leif Hammerstrom at Uppsala University has actually now seen spectroscopic evidence of this LEPT state in these triads that do not show inverted region behavior. So we have a better understanding of how this whole process works. Now I want to talk about a biological example. As my last example, this BLUF photoreceptor protein. BLUF stands for blue light using flavin. So this flavin absorbs blue light, and that leads to a PCET reaction, which then can lead to conformational changes in the protein, which then can send a signal 50 angstroms away to turn on or off an effector. So BLUF coupled to an effector can cause changes under blue light. It can cause neurons to fire, reactions to be catalyzed, and so forth. And this is relevant to optogenetics, where light is used to manipulate cells in living tissue with very high spatial and temporal resolution. You can see this poor rat, it has this probe in its brain, right? When you shine blue light on this probe, you can get the rat to do certain things because of this sort of a bluff photoreceptor. Now, we don't want any rats in our lab. I certainly don't want any rats when I'm working at home during the pandemic. So instead, we're actually using this as a model for protein modulated photoinduced PCET. So that's what I'm interested in using this protein for. So in the photocycle, we start out in step 1, shining blue light on the flavin to photoexcite the flavin to a locally excited state we'll call FMN star. The second step is electron transfer from tyrosine to the flavin, forming the FMN anionic radical. The third step is proton transfer from tyrosine to the flavin through this glutamine, forming the FMNH neutral radical. And then the fourth step is a little bit murky. They don't really know how it works, but there's some sort of reverse PCET that leads to the flavin and the tyrosine going back to their initial oxidation and protonation states. And then it's in some sort of a thermally excited light adapted state. This light adapted state is able to turn on or off the effector. And it can last up to 5 seconds before eventually it thermally relaxes to the dark adapted state. So what we want to do is we want to figure out, can we understand the different steps, the forward and reverse PCET reactions? And can we understand what the light adapted state is? What makes it different from the dark adapted state? Because these are unknown questions. They couldn't see them experimentally. They were able to see these radical intermediates. So they know these steps in the forward direction, but after that, everything gets a little bit confusing. So to do this, we did QMMM molecular dynamics simulation. So all of these atoms shown explicitly were in the QM region, the quantum mechanical region. We equilibrated on the ground state. We photoexcited to this locally excited state of the flavin, and then we did non-equilibrium on-the-fly TD-DFT dynamics. We observe electron transfer from tyrosine to the flavin. We observe the double proton transfer. Then we see a rotation of this glutamine, and then we see reverse PCET. So I'm going to show a movie now, where we start out with photoexciting the flavin. Now you can see this proton transfer, and you can see this rotation of the glutamine. And then eventually we're going to see the reverse proton transfer. You don't see the electron transfer steps because you're not going to see them in the according to where the atoms are. There you go, reverse proton transfer. So what we found is we were able to understand how protein reorganization and electrostatics could induce the electron transfer reaction, but more importantly, we could characterize the light adapted state. You see this hydrogen bonding interaction between the this glutamine tautomer and this CO. That will lead to redshifts of the electronic absorption in the CO mode, which is characteristic of the light adapted state. So to show it, it's hard to see in the movie because it happens so fast. So we start with the dark adapted state with just the regular glutamine. We see electron transfer and proton transfer. That's forward PCET. Then we saw this glutamine rotate. And then you see the reverse PCET reaction, which is only possible because of this rotation of the glutamine. And then you see now we've got this hydrogen bonding interaction between the imidic acid glutamine tautomer and the CO of the flavin. And this glutamine tautomer is very high energy. It's rarely seen in biology, but in this case, you can see it's stabilized by hydrogen bonding interactions. And it also only occurred because of this photocycle. It's an unusual situation. And what we could do is we can actually calculate the electronic absorption of the flavin and the IR vibrational stretch here of the CO for both the light adapted state and the dark adapted state before anything started. And we see these red shifts in both of these spectra. And these red shifts agree with experiment. So we can't prove that this is the light adapted state, but at least we have evidence that it has the characteristics of the light adapted state. Now, in this talk, I started out talking a lot about the analytical rate constant expressions and how important treating the hydrogen nucleus quantum mechanically and including excited electron proton vibronic states is for understanding a lot of the systems that we studied. And then I also looked at non-equilibrium dynamics, and in these non-equilibrium dynamic simulations, all of the nuclei were treated classically, which turns out to be okay for these cases, but there's a bit of a disconnect. So what we really want to be able to do is we want to be able to do dynamics with quantum nuclei for these photoexcited reactions. Look at the non-equilibrium dynamics. And for this reason, we're developing the nuclear electronic orbital, or NEO method, where we treat specified nuclei quantum mechanically on the same level as the electrons. So for example, for H5O2+, we could treat all 5 protons quantum. The 2 oxygens are just regular point charges. And we've developed NEO-DFT, NEO-TD-DFT to get excited states, both electronic and proton vibrational and mixed electron-proton vibronic excited states. We've developed wave function-based methods, so coupled cluster singles and doubles, the EOM method to get excited states with the wave function methods. And all of these methods include nuclear quantum effects and non-Born Oppenheimer effects into quantum chemistry calculations in a computationally practical way to allow us to calculate pKa's, geometries, reaction paths, tunneling splittings, vibronic couplings, isotope effects, and so forth. For the purpose of this talk, I think what's most interesting is that we've more recently started to look at real-time neo-TDDFT with Aaron Phys dynamics. And this is a collaboration with Xiaosong Li's group. Luning Zhao is the first author on this paper, where we're now integrating the nuclear and electronic dynamics in a time-dependent framework. So we can look at dynamics beyond the Born-Oppenheimer approximation. We can resolve nuclear and electronic spectral features, but more importantly, we can look at the non-equilibrium dynamics of photoinduced intramolecular proton transfer, or PCET. So in this example, we photoexcited this molecule to an excited electronic state, and we could look at the electrons and the proton as they evolved in time. And that's what's shown here in this movie. The green and the blue are the electron densities, and then this proton, this white blob, is the proton, and they're all coupled to each other without any Born-Oppenheimer separation between them, and we can understand then what's going on. And this is, in this case, all of the other nuclei are fixed, but then we can do Aaron Phys dynamics and move the other nuclei as well. So we're developing this kind of method so we can look at PCET reactions, including these nuclear quantum effects and the non-Born Oppenheimer effects. So to summarize, we've developed a general theory for PCET. We've derived rate constant expressions for PCET in solution, proteins, and electrochemistry. We've identified important aspects of PCET. We see hydrogen tunneling and contributions from excited vibronic states are important. We see that we can tune these reactions with substituents that impact the thermodynamics. We see the kinetic isotope effect can range from 1 to more than 100, and we see the Marcus inverted region behavior is possible. We've also looked at the photoinduced PCET reactions, and we see the importance of non-equilibrium dynamics, protein solvent reorganization, and electrostatics. So all of this we also want to start studying with this NEO method and try to understand better how the dynamics is really related to some of the experimental phenomena. So of course, I couldn't do any of this without my group, so I'm very indebted to all of them. Alexander Sudikov has been working on the PCET projects for many years, so he's essential. Moy Quinn and Josh Goings worked on the BIP molecules. Zach Goldsmith and Jan Choi Lam and Alexander also worked on the heterogeneous PCET, the Volmer reaction. And Zach Goldsmith also worked on the inverted region work, and Elvira Seyfuchirova worked on the non-equilibrium dynamics of those triads. And Josh Goings did the work on the bluff system that I showed at the end. And all of these people have contributed to the NEO, including John Tully, who has been wonderful in providing his wisdom. And the NEO method's been going into these different codes here. And of course, we're also indebted to our theory collaborators on the real-time NEO methods, Xiaosong Li and his students and postdocs, and our experimental collaborators who keep us working on interesting problems, and these places for funding. So I'd be happy to answer any questions. Thank you very much.
That's great, Sharon. One of the unfortunate things about this format is that you don't get to hear a giant room of people clapping for you, but you just have to imagine. So as we said, we're going to have Rodrigo Noriega and Carlos Baiz alternate to moderate the questions. And so you can go ahead and get your questions into the chat feature, and Rodrigo Noriega from the University of Utah is going to take the first one for you.
Hi, Sharon. Thank you for such an interesting talk. The first question is related to the observation of the inverted region. So because observing the inverted region depends on the overlap and the activation barrier, does reducing the spacing between vibrational states make it less likely to observe the inverted region by having more excited states within the nearly activationless energy slice? Or is that not an important factor because of the highly oscillatory nature of those states?
Yeah, that's a really good question, and it's actually a subtle interplay between the splittings are, the short answer is the splittings are important. It's one factor that can be relevant. It's also the shape of the proton potential. So for example, these highly oscillatory states have to be above the barrier. So the barrier has to be low enough that these states are above it, and so that the oscillations are going to lead to this phase cancellation. We actually did a very detailed study in this Faraday discussion from 2019, where we looked at model systems. And so we tried to answer exactly that question as to which factors are important. And it turns out there's not a straight, simple answer. But certainly the splittings are involved in determining, because it is this balance between overlaps and free energy barriers. And of course, in this case, the Boltzmann population is not as important because they're pretty much all starting in the ground state. But this delicate balance between free energy barrier and overlap, it's hard to predict ahead of time without actually just going ahead and doing the calculation and plugging into the rate constant expression. And then you can see which states are dominant. But again, it's really having a proton potential that's with a low enough, an asymmetric proton potential with a low enough barrier. It can't be a single well either. It has to be asymmetric double well with a low enough barrier such that the excited states that would be activationless are delocalized above that barrier. That tends to be the case when you see this kind of quenching of the overlap, which means that the excited states can't contribute, which means you will see inverted region behavior.
Thank you.
And I guess I'll ask the next question. Sharon, thank you for the wonderful talk. There's the question in the chat that says, in an electrochemical proton couple, the electron transfer, whether it's homo or heterogeneous, if the process is occurring with an applied bias, which I think you mentioned at the beginning in your theory, can you account for time-varying potentials in that applied bias?
Yeah, that is a good question. We have not looked at time varying so much. So, I mean, you can do, we have simulated CVs. So in that sense, a very, time varying as in a sweep of the potential. We did some work in collaboration with Joseph Otnick's group on PCET electrochemical interfaces. So there the only time variation we've done is really in this sort of linear sweep that we did in that to just generate a CV. But we have certainly looked at the effects of just the applied potential in general. It does affect the electric field at the interface. And so we've been very interested in understanding how the electrostatics at the interface can drive PCET. And so we've been more concerned about that than about the time dependence. So we figure we should first understand how it is just in a single time, really understanding how because the fields turn out to be very inhomogeneous at the electrode solution interface. And so understanding those inhomogeneous fields and how they change with applied potential is something that we're spending a lot of time thinking about and working on. And I think once we get a better handle on that, a better understanding of that, then we could maybe add the time dependence to that. Other than, like I said, that's sort of more straightforward, just linear sweep that you would do for a CV.
Great. Thank you. And I guess Rodrigo will take the next question.
Yes, we have the next questions ready from the chat. This question comes from Lu Wang. First of all, they thank you for a great talk. And then the question is related to the bluff system. It seems like forming the hydrogen bonds with the proper length is crucial for the proton transfer. Is the proton transfer rate strongly dependent on the hydrogen bond length? And is the formation of the hydrogen bonds modulated by the protein structural fluctuations? Could you comment on the timescale for the rotation of the residues and the formation of hydrogen bonds?
Yeah, so this is a good question. So you're absolutely right that having this network formed is really important. And we've actually done simulations, free energy simulations of the formation of this proton wire, this hydrogen bonding network. And the protein does thermodynamically favor the formation of this. Certainly in the locally excited state, we found that it was favored. Depending on the bluff species, we've looked at multiple species, 2 different species. It may be more favored in one than in the other. For example, this species I'm showing here, it is highly favored to have this proton relay formed. And so that is an essential part of getting this to work. In terms of time, so in some cases it's just already formed. There's no time scale associated with it. It's pretty much there. Like I said, in the APA bluff, which is this is the SLR bluff, which is where it's pretty much always formed. In the APA bluff, sometimes it's not formed in the ground state, but when you photo excite it, it becomes thermodynamically favored to have it form. So I don't think there's much of a timescale associated with the formation of those hydrogen bonds, but in terms of the different steps, like here we go. This, for example, we know experimentally that this species stays around for about 7 picoseconds, this anionic radical. The neutral radical stays around 6 picoseconds, and this before the reverse PCET stays around 65 picoseconds. But that's from experiments, not from our theory. From our theory, it's hard. What we had to do, in fact, to get the reverse PCET is we couldn't wait 65 picoseconds doing QMMM for this sort of a large system. So instead, we actually had to enforce the electron transfer to see the reverse reaction. So in some sense, we had to force it to happen faster because we couldn't wait that long. But we do know all those timescales. Like I said, these are like 6 or 7 picoseconds for these 2, And then this one lasts on the order of 65, 70 picoseconds. Sorry, these are picoseconds. And so, so that is known. And then the light adapted state can last up to 5 seconds. So those are the timescales involved.
Thank you. Carlos, you want to take the next one?
Yeah, the next question is really interesting. It's talking about the activation-less photoinduced PCET, whether you see computational evidence for transferring from, basically climbing down the vibrational ladder after you make the transfer. Do you see that relaxation in the computations?
Yeah, no, that's a good question. I'm going backwards. Yeah. Do we see the vibrational relaxation you're saying? So we're in the 0-3 state, and it's going to, it's going to relax to the ground state. So in our theory, we don't, that doesn't include that part, right? Once we get the rate constant, once it's transferred, that we get the rate constant, we don't look at that relaxation. In principle, if you were doing some sort of non-equilibrium dynamics, say with a method that does include nuclear quantum effects, say once our NEO method could look at these kinds of systems, you might be able to actually see that relaxation. But we have not yet. You really need a method that's going to, you have to include the nuclear quantum effects if you're going to see that kind of vibrational relaxation. But I think it happens fast enough that the experimentally, right, from their perspective, right, it's already usually when they're going to see it when they probe it in the ground state. Yeah, so that's something we really do want to look at. And we're hoping some of these new methods that we're developing will allow us to look at that level of sort of vibrationally excited states during the actual reaction. But the theory, the analytical rate constant expression does not include that subsequent relaxation.
Thank you.
So the next, I'm grouping two questions from the chat. Well, one from YouTube and one from the chat that kind of go more to the revisiting of some of the basic concepts that you described at the beginning of your talk, Sharon. So the first one comes from Jessica Santos, and she asks whether the PCET rate constant that you derived serves for all mechanisms or simply for the concerted mechanism. And the second part of the question comes from Rob Weekly, asking, given the relative rate of motion for electrons and protons, is it possible to differentiate concerted motion and proton motion followed by electron motion?
Okay, so the first question, the theory that presented here is for concerted. It's assuming concerted PCET. We also have expressions, go back to this. We also do have expressions for, there it is. So this expression here is assuming concerted PCET. We do have expressions, if you have sequential PCET, if you know there's a thermodynamically stable intermediate, then we have expressions for those reactions, but it's really just going to be using the single electron and single proton transfer expressions sequentially, so one after the other, so depending how coupled they are. So those expressions, we have a chemical review with Alexei Shcherbukov that has those expressions in it, but they're fairly standard expressions, sometimes from the literature, where you can, so the sequential ones are fairly straightforward to deal with as well. If you know there's a thermodynamically stable intermediate, you can just use the sequential ET and PT one after the other. You have to be sure you use the right limit, adiabatic versus non-adiabatic. So you have to use the right rate constant expression for electron transfer and proton transfer. But other than that, we have this sort of framework to do that as well. But this particular expression is assuming concerted PCET. The second question about timescales, I mean, that gets a little tricky because in the non-adiabatic regime, right, the timescales, first of all, electrons and protons are both quantum in this description, so that even the definition of what you mean by timescale becomes confusing, right? Because they're wave functions, they're delocalized. And so talking about the timescale of an electron and a proton becomes a little ill-defined. We can define effective timescales using a semi-classical formalism. And so we can, for example, get an electronic transition time, and we can get like sort of a Landau-Zener kind of approach, an electronic transition time and a proton tunneling time. And we do calculate those for different systems, and we can see when you can see whether the proton tunneling time or the electronic transition time is faster. And that tells you whether you're in the non-adiabatic or adiabatic regime. This is what I mean here on the slide, actually, by electron-proton non-adiabaticity has to do with the relative timescales that you can in fact compute with the semi-classical formalism. But again, it's you have to be careful about talking about timescales because they're so coupled to each other, the electron and proton, there's no Born-Oppenheimer separation between them. It's really hard to talk about the individual electron and proton transfer time. So I tend to try to just really say they're just electron-proton vibronic states that we're dealing with and not try to kind of separate them too much, at least for the concerted PCET mechanism. But these are all very deep questions that we have thought about. We have, papers on this about this sort of semi-classical formalism and trying to understand the different limits. But again, it becomes a little bit of a semantic issue. And in this particular theory, we're really just talking about sets of electron-proton vibronic states.
Great. And the last question from the chat comes from one of the participants. It says, is it possible to modulate the electron self-exchange reaction, model the electron self-exchange reaction as PCET if you consider vibronic coupling? I think the idea is if you could, make an excited state, right, and then you have some nuclear motion, can you treat that as PCET?
So in general or for a specific system? I'm not sure I fully understand the question, but certainly you can. So for example, the proton donor acceptor motion that we're talking about here, you can include a mode within the molecule in your PCET theory. In fact, we do include this specific mode. If there was another mode that you think modulates the PCET reaction, you could also include that in your theory in a similar, analogous way. I'm not sure that's really what the question was, but
yeah, I'm not sure I understood it either, but thank you.
That's what I got. That's what I got from it.
Yeah,
so
right.
Right. If there's a critical mode that you want to include in your, and that really is important for PCET, whether it's the electron part or the proton part, then you can include it the same way we included this proton donor acceptor mode R. You can do some sort of a thermal averaging procedure, assuming it's at equilibrium, right? Which is what this is assuming. You could, in principle, just include it in the same analogous way. And so that would certainly, you could model that if you had evidence of a specific mode you wanted to include.
Thank you. I think that was the last question.
Yeah, I think it was. That was great. Again, Sharon, an excellent talk. I thought those were fun questions, and thank you for taking the time to answer them. What we'd like to do now is kind of two things. Thank all the participants, of course, for logging in, and also to say that it's time to be thinking about future speakers for this series. And so please be looking within the next week at the Phys Division's website for a poll where you can suggest future speakers for the series. Now, if you're not a member of the Physical Chemistry Division, you need to join the Physical Chemistry Division so you can get an email where you have a chance to give feedback, and you'll be able to keep up to date on who the next speaker. So make sure to do that if you haven't. And the other thing that needs to happen now is that all of the faculty, aside from Professor Hammes-Schiffer, among the attendees need to go away and have a happy hour because now is the time for the grad students and the postdocs to have